Hello everyone, welcome to sqadia.com. Today we are at the discussion about the cellular energetics, and this section we are going to start off with the discussion of metabolism of glucose, and we are going to start off initially by the discussion of getting an overview of how energy is being catered either by the production of the glucose and how after the glucose has been produced, take an example from the photosynthesis, is being utilized in the formation of energy. So we are going to start off with the generalization and the utilization of the proton motive force, and we are going to see how that proton motive force, which is basically a combination of both the electrochemical gradient and the proton gradient, is being produced and how it is being utilized in several different processes. Then we are going to see the detailed structure of the processes that are involved in the glycolytic pathways. And lastly, we are going to start off with the discussion of the difference between the aerobic and anaerobic metabolism of glucose. So, starting off with the discussion of the utilization of the proton motive force, what we have over here, we have the generation of the proton motive force that is primarily because of the two reasons. Number one is the chemical energy, which is being produced from the mitochondria and the aerobic bacteria. The second is the light energy, and this light energy is basically hinting towards the process of what we know as the photosynthesis that occurs in the chloroplast and in the photosynthetic bacteria as well. So both of these combined, they are the combination of both of these chemical and the light energy is catered under the definition of the proton motive force, where the proton gradient and both the electrochemical gradient is being utilized in it. So what happens, this particular diagram shows that there is the concentration gradient from outside to the inside, and of course this department of course is sealed, where is the there is an electric potential and there is a hydrogen concentration gradient. Both of these combined are going to serve as the proton motive force. Now we have the exoplasmic phase over here and the cytosolic phase over here. So the energy that is being produced in the form of ATP during the synthesis of the ATP from ATP synthase that occurs in the mitochondria and in the aerobic bacteria. So what happens, this energy is being utilized in the chemiosmotic coupling. It means that the chemiosmotic coupling, as the name indicates, it is basically the transport of a molecule against the concentration gradient. And there are two different types of transport because we are talking about coupling. So there either would be a symport in which two molecules are going to move into the same direction from the exoplasmic phase towards the cytosolic phase, or we are talking about the antipode in which one of the molecule is going to get out from the cytosolic phase into the exoplasmic phase, while the other molecule, take an example of the hydrogen ions, they are going to move from outside to the inside. This ATP synthesis can also play its role in the rotation of bacterial flagella. That plays an important function if we are talking about the pathogenesis to move down through the, of course, the tract, the urine. Either we are talking about the tract, which plays an important function in the establishment of pathogenesis of many bacteria. So moving on towards the glycolytic pathways, we have the starting molecules, the starter molecule, the glucose. It is being converted into the glucose 6-phosphate, of course, by the addition of a phosphate group, and that is done in the presence of a hexokinase, which of course is a kinase. If you know, all kinases are going to cause the addition of the phosphorylation, if you can recall from the cell signaling processes. Of course, this process would also require the use of ATP as a source of energy. So for glucose 6-phosphate is converted into its isomer form, that is of course the fructose 6-phosphate, through an enzyme, the phosphoglucose isomerase. This fructose 6-phosphate is then going to be converted by the addition of another phosphate group into fructose 1,6-bisphosphate, which is then going to be converted into two different processes depending upon the enzyme that is acting on it. So in the presence of the aldolase, it is going to be converted into dihydroxyacetone phosphate, as shown over here by this, and this reaction of course is reversible. And this can also be converted into the glyceraldehyde 3-phosphate or the G3P molecules through by the action of the glyceraldehyde 3-phosphate dehydrogenase, of course. So the next step for the G3P, of course, is the action of the glyceraldehyde 3-phosphate dehydrogenase, and this enzyme is going to act on the glyceraldehyde 3-phosphate and two molecules of the glyceraldehyde 3-phosphate, because it's important to consider that G3P is a 3-molecule structure, is a 3-molecule compound, while glucose was 6 carbon compounds. So over here G3P contains 3 carbons, the glucose was containing, of course, the 6 carbons. So, of course, its action, the glyceraldehyde 3-phosphate dehydrogenase action, would produce 1,3-bisphosphoglycerate, which would be converted, of course, by the removal of the phosphate group to 3-phosphoglycerate molecule by the action of the phosphoglycerate kinase, and of course this would be converted into the 2-phosphoglycerate molecule by the phosphoglyceromutase enzyme. Now, when this has been produced, this is going to be converted into phosphoenol pyruvate, also known as PEP, by the action of enolase. And all of these processes are reversible. So it is entirely upon the abundance of a particular compound and of course the enzyme, subsequent enzyme, that drives the reaction in either way. Now, phosphoenol pyruvate, when have been synthesized by the addition of 2 subsequent ATP, because these are going to cause the production of the ATP through the pyruvate kinase, and they are going to be converted into the pyruvate. So 4 molecules of ATP have been generated, 2 molecules at step 7 and 10, 2 molecules at 10, while the NADH have been produced as well. So, talking about the anaerobic versus aerobic metabolism of glucose, we have 3 examples in front of us. First is the yeast cytosol, the second is the muscle cytosol, and third is the normal cell cytosol. So, let's start off with the yeast cytosol. What happens in this case? The glucose is going to be converted by the process of the glycolysis into the pyruvic acid, which we have just established in the previous glycolytic pathways. That glucose, the ultimate fate of the glucose at the intermediary product, is the pyruvic acid or the pyruvate. Now, this pyruvate, in case of yeast, because all of these, the yeast, muscle cytosol or the normal cell cytosol, all of these are going to produce the pyruvic acid as a process of the glycolysis through a pathway that we just discussed, the glycolytic pathways, causing the production of 4 ATPs and 2 NADH. So, what happens in case of yeast, pyruvate decarboxylase is going to act on the pyruvic acid. Now, it's a decarboxylase. It means that it is going to cause the decarboxylation. It means that it is going to cause the removal of the carbon in the form of carbon dioxide. So from the pyruvic acid, when the carbon dioxide has been removed, as shown from this particular figure, that this CO and this O has been removed, and the resultant compound would be acetaldehyde. This acetaldehyde is going to act upon by an enzyme called dehydrogenase, which is then going to cause the donation of one of the, of course, hydrogen. This is going to cause the production of from the acetaldehyde into the ethanol, and this process is termed as alcoholic fermentation. Talking about the muscle cytosol, when we have the muscle fatigue, there is a lactic acid fermentation in it, and this is the process that basically is acting because there is no abundant amount of oxygen that a muscle can carry at that particular level where there is an extraneous exercise involvement. Because of the absence of the oxygen, the pyruvic acid, which has been metabolized from the glucose in the absence of the oxygen, is going to be acted upon from the lactate dehydrogenase and would be converted into lactic acid. And of course, it's important to consider that 1/4 of the lactic acid is not converted back to the pyruvic acid, but the 4/5 of the lactic acid has been converted after the oxygen is being supplied, of course. Talking about the cytosol, the pyruvic acid is then being transferred to the mitochondria, where pyruvic acid is going to be converted into the acetyl CoA, and we are going to see how this process is going to be occurring. in the coming sections. Acetyl-CoA would undergo the citric acid cycle, and of course there would be oxidative phosphorylation, and overall it is going to be converted into the end product, which would be the release of the two carbon dioxide molecules, which is then going to enter the NADH produced, the FADH produced is going to get entered into the electron transport chain, and of course they are going to cause the production of ATP by the movement of electrons, which are going to lose their energy, and this loss of energy would be given to the proton pumps to move against the concentration gradient, so that these protons are accumulated outside, and when they move inside, with the help of some channels that are present in the ATPase, the ATP synthesis would start. So in this section, we started off with the general discussion and the utilization of the proton motive force, and then we moved towards the glycolytic pathway, and lastly we discussed the anaerobic versus aerobic metabolism of glucose and their end product. So that was all for this section. Thank you for watching sqadia.com, and we'll see you in the next section.